Now, as I mentioned before, the stages of transcription are initiation, elongation, and termination. It might be helpful at this point to go to this part in your textbook and look at the pictures as we talk about it. With initiation, the promoter of the gene includes the transcription start point. This is the nucleotide where RNA polymerase begins the messenger RNA synthesis and extends tens of nucleotides upstream. The RNA polymerase binds directly to, at a precise location and orientation on the promoter. And this is going to determine where transcription starts and which of the DNA strands is the template. So then the DNA strands unwind. Excuse me. The eukaryotic promoter usually includes what we call a TATA box, which is a nucleotide sequence with just TA, TA, TA. 25 nucleotides or so upstream from the start point. There are several transcription factors. One recognizes the TATA box and must bind to DNA before RNA polymerase II can bind in its correct position or orientation. Excuse me. Additional transcription factors bind to the DNA along with the RNA polymerase II, and this forms transcription initiation complex. And these are all protein-protein interactions. The RNA polymerase II unwinds the DNA double helix, and RNA synthesis begins at the start point on the template strand. Now, second part, elongation. In bacteria, polymerase moves downstream, unwinds the DNA, elongates the RNA 5' to 3', and then afterwards the DNA reforms its double helix. In eukaryotes, RNA polymerase untwists the DNA, 10 or 20 nucleotides at a time, and adds RNA nucleotides to the 3' end. Once formed, it peels away and the DNA reforms. Transcription in eukaryotes occurs about 40 nucleotides per second. A single gene can be transcribed simultaneously by several molecules of RNA polymerase that are lined up. These growing strands encode protein in very, very large amounts. Finally, termination. In bacteria, the RNA transcript is released, the polymerase detaches from DNA, and this proceeds through a terminator sequence in DNA. The transcribed terminator functions as a termination signal, and thereby the transcript is released. No further modification is required. No processing occurs. In eukaryotes, RNA polymerase II transcribes on the DNA what's called a polyadenylation signal sequence, and this specifies a poly, which means many, of course, adenylation signal, which will look like AAU, AAAAAA, N, pre-messenger RNA. This is called a signal because as soon as the 6 nucleotides appear, the AAU, AAA, they're immediately bound by certain proteins in the nucleus. 10 to 35 nucleotides downstream from the AAU, AAA, the proteins cut the RNA transcript free from polymerase. This releases your pre-messenger RNA, and then it can be processed. RNA polymerase II can continue to transcribe. The enzyme begins to degrade the RNA starting at the newly exposed 5' end. Polymerase continues transcribing, pursued by enzymes, catches up to polymerase, and dissociates from DNA. In eukaryotic cells, we modify the RNA after transcription. This is done by enzymes. It's called RNA processing, and both ends of the primary transcript are altered. At the 5' end, we get a 5' cap, which is a modified form of a guanine nucleotide. It's added after the first 20 to 40 nucleotides. The 3' end, the enzyme adds somewhere between 50 and 250 adenines, and this is called a poly-A tail. And you might wonder why. Well, this facilitates the export of the mature messenger RNA from the nucleus via the nuclear pore. It protects messenger RNA from degradation by hydrolytic enzymes, and it also helps the ribosomes attach to the 5' end of the messenger RNA once it moves to the cytoplasm. There are untranslated regions. They're called UTRs. They're the 5' and the 3' UTR, and these are parts of the messenger RNA not translated into protein, but they have other functions like binding of the ribosome. Now, as RNA is processed, RNA splicing happens where large portions of RNA molecules are removed and the remaining parts are reconnected. The average length of a transcription unit in humans is 27,000 nuclear pairs. The average size protein is about 40 amino acids. So if you multiply that times 3, that requires 1,200 nucleotides in the RNA code. Introns stand for intervening sequences, and these are non-coding segments that lie between coding regions. And what we do, we know that these have to be removed. Exons are those that are expressed, and they're usually translated into amino acid sequences, with the exceptions being the UTRs. The terms intron and exon are used for RNA and DNA sequences that specify them. Introns are cut out, exons are joined together, and this forms messenger RNA with a continuous coding sequence. Introns can be removed by a complex of proteins and small RNAs called a spliceosome. It binds to both ends and sequences along the intron. It is released and then degraded, and the spliceosome joins the two exons. The small RNAs in the spliceome participate in spliceosome assembly, in splice site recognition, and they help to catalyze the splicing reaction. Ribozymes are RNA molecules that function as enzymes. In some organisms, RNA splicing occurs without proteins or additional RNA molecules. The intron RNA actually then is functioning as a ribozyme and catalyzes its own excision, which means to cut and remove. This is important because the discovery of ribozymes disproved that all biological catalysts are proteins. There are 3 properties of RNA that allow it to function as enzyme. RNA is single-stranded. The region of RNA molecule may base pair with a complementary region elsewhere in the same molecule, and this gives a molecule a particular 3-dimensional structure. And this is very, very important, this structure, for the catalytic function of ribozymes. Certain bases in RNA have functional groups that participate in catalysis, and the ability of RNA to hydrogen bond with other nucleic acid molecules, whether they're RNA or DNA, adds specificity to the way it operates as a catalyst. So what are some of the functional and evolutionary importances of introns? Well, first of all, RNA splicing, the presence of introns, does it give evolutionary advantages? Some of these contain sequences that regulate gene expression. Many affect gene products. It allows one gene to encode for more than one kind of polypeptide, and this is called alternative RNA splicing. It's one reason that we have such a low number of genes, because the number of products that can be formed is much greater than the number of genes that exist. Proteins have modular, structural, specifically functional regions called domains. Different exons code for different domains. Exon shuffling is when the presence of introns in a gene can facilitate the evolution of a new, potentially beneficial protein. Introns increase the probability of crossing over between exons, and now we'll be talking about translation.